what is a example of negative feedback. Now, let's say you are out in the cold and you start shivering. Okay, let me pull that thing here. Okay. Now, in this mechanism, if you're out in the cold and you start shivering, what is the effector organ? Like we said, there are three components. You have the receptor, you have the control center, and then we have the effector organ. Receptors are always, if it is the body, the temperature changing, so you have those temperature receptors in the body. Brain is the control center always. Now, what is the effector organ when you're out in the cold?
Your muscles.
Yes. So you should be able to answer questions like this. What is the effector organ if you're shivering or if you're out in the cold? Yes, it is the muscles. Now, on the other hand, if you're out, you're overheated and then you start sweating to cool down the body temperature. Now, what is the effector organ in that case? What is producing the sweat?
Sweat glands.
Yes. So the sweat glands would be the effector organ. Okay, any other examples of negative feedback? Give lots and lots of examples.
Like insulin to lower your glucose levels.
Yes. So maintaining your normal blood sugar level is another example of negative feedback. Now, what is positive feedback?
Childbirth.
Okay, that's one example of positive feedback. Any other example? Related to childbirth?
Breastfeeding.
Breastfeeding. And then there's another one we talked about, blood clotting. So know the examples of each one. Okay, so we're done with. Okay. Now, scientific method. What are the different steps in the scientific method? The first thing is
observation.
Yes, you observe something and then you go back and you do all the research on whatever you think is the question that came to your mind based on that observation. So what do you create? Hypothesis. So just know these steps, and this was another question in one of the worksheets you did. So you have the hypothesis, and then what do you do next after that? After you've created the hypothesis, what do you do?
Yeah. Experiment.
You do the experiment. And then what is the end? Here, accept
results.
So whatever was your hypothesis, either you accept or you reject, or if the results were not consistent, then you have to modify and do it again. So just know what are the basic steps of the scientific method. And likewise, what are the differences between science, non-science, and pseudoscience? What is science?
It follows the scientific method.
Follows.
Yes, something which follows the scientific method, like what we just talked about. And it is always peer reviewed. So that's why it is got a lot of backing, because it's not that you just come up with something. It has gone through a lot of different steps before you start following it. So it's science. Now, what is non-science? Example.
Religious beliefs.
Yeah. There's no scientific method. It's just your belief, and that's what you follow. So that is non-science. And what is pseudoscience?
Non-science that acts like science.
Yeah. Any example you can remember of pseudoscience?
Astrology?
Yes. Telepathy? Yes, that's all pseudoscience. Okay, so that's all in this chapter. Any questions before I pull up my next PowerPoint? Okay, I'll pull up my second one. You might want to know the structure of an atom. I don't think I need to go through that. You know that the structure of the atom, what is a proton, what is a neutron, and electron, and what is atomic number and atomic mass. So atomic number is based on the number of protons, and a mass, atomic mass is the mass of protons and the neutrons together. So you might want to know those definitions. What is isotope? What is different in the isotope?
The amount of neutrons.
Yes, it's the number of neutrons which is different, and neutrons are actually neutral, so the charge does not change, but because of the neutrons, it makes it unstable. So that's why you have carbon 12, 13, 14. That's one example here, and that's what these isotopes are used in medicine a lot. You know, all the radiotherapy and even the gamma knife and all, lots of it is used in medicine. So that is what is isotope. So know what is isotope. Okay, then the next thing would be the types of compounds, ionic and molecular. So what is the ionic compound? What connects the different ions? What kind of bond is present?
Ionic bonds.
Okay, so what happens in ionic bond? Are you sharing the electrons, or are you donating and accepting the electrons?
Sharing them.
Sharing would be covalent. Ionic is one is donating and the other one is receiving. That's why they become ions, like sodium chloride. By itself, sodium chloride is neutral, it's a salt, but when you dissolve it in water, it breaks down. What happens? Sodium has donated its electron to chloride, so sodium becomes positively charged, and chloride, which accepts the electron, becomes negatively charged. So the kind of bond between these two is the ionic bond, where one donates and the other one receives. Molecular compounds are where you have the electrons which are shared. So that is what is that is covalent bonds. Okay, what are cations? What charge do they have? Positive. Yes, positive. So sodium with a plus sign, it's a cation. Calcium with 2 plus is a cation. And what is an ion?
Negative.
Negative. Example. Which one gets a negative charge?
Chlorine.
Yeah. Yep, you're right. So that is an anion. And here you have all these listed here. Okay, so what are isomers? Like we talked about isotopes, what are isomers?
Molecules that have the same molecular formula but different structures.
Yes, different structures. Like here you see the example, glucose, galactose, fructose. They all have the same formula, C6H12O6. But because the arrangement of all these are different, so that's why the structural formula is different, and these are called isomers. So know these definitions. We said covalent bond is when the electrons are shared. Okay, the next thing, yes. What are polar and what are non-polar covalent bonds? What are non-polar covalent bonds?
When two atoms have the same electronegativity.
Sorry, say that again, please. When two? They have the same.
Two atoms have the same electronegativity.
Okay, how about the sharing of electrons?
Equally.
Equal. That's why they have same electronegativity, because they are sharing the electrons equally. And that is non-polar. And what is polar? When the electrons are not shared equally. There is unequal sharing. Like in water, oxygen will pull the electrons more towards itself. So the electron is closer to oxygen, so oxygen will get slight negative charge, whereas hydrogen will get a slight positive charge because that electron is further away from hydrogen. So that is what is the polar covalent bond. So you might want to read about polar and non-polar covalent bonds. And then what are amphipathic molecules? What are amphipathic?
Is it where they have like a polar and then a non-polar space?
Yes, they have both. One molecule will have both polar and non-polar molecules, and that's why it is not molecules, polar and non-polar regions in one molecule, and that is amphipathic. What is the example? We've talked a lot about that. What is one example of amphipathic in the cell?
A phospholipid.
Yes, the phospholipid bilayer. So the plasma membrane, which has phospholipids, phospholipid is amphipathic. So it has the polar head, which is hydrophilic, and the tail is hydrophobic. That means it is non-polar. Okay, so you know the bonds now. What is surface tension?
Inward pulling of cohesive forces at the surface.
Yes, and that is important to the body because this surface tension tends to collapse the lungs, but then we have a chemical called surfactant which keeps the alveoli open. So you might want to know about surface tension. Okay. And here you have, so summary, polar molecules dissolve in water because they are hydrophilic, they like water, so they can dissolve in water, whereas non-polar will not dissolve in water. Okay, so we've talked about I think that's all in this one. Any questions? Okay, so let me pull my next PowerPoint. Okay, so in terms of the pH, if a solution has a pH of 4, is it acid or a base or neutral?
Acid.
Yes. So acid would be anything pH less than 7 would be an acid. And if the pH is 10, is it acid, base, or what?
Basic.
Base.
It will be a base. Now, moving on to in terms of, okay, let's first talk about, let's say pH of 2 and 3. Which one would be a stronger acid? pH of 2 or pH of 3?
2.
pH of 2, yes, smaller the number because the strength of the acid will increase towards 0. And comparing base, at 10 and 11, which one would be a stronger base?
11.
11. Now, in terms of hydrogen ions, when you're comparing pH of 2 and 3, which one will have more hydrogen ions?
2.
2, yes. 2, because stronger the acid, more are the hydrogen ions. So pH of 2 will have more hydrogen ions compared to pH of 3. Okay, now going on to the biological macromolecules, lipids. What are the monomers of lipids?
Fatty acids.
Fatty acids. And what is the function of lipids? It's all listed here actually.
Energy storage, compose cell membrane, and create hormones.
Yes, good. Okay, and you might want to know the classes of lipids. So triglyceride, phospholipids, steroids, and eicosanoids. These are the 4 classes of lipids. Okay. Note these definitions, what is lipogenesis and what is lipolysis. So what is lipogenesis?
Triglyceride formation occurring when there are extra nutrients.
Yes, so formation of the lipids. And lipolysis would be the opposite, breakdown. You know, lysis means breakdown. So breaking down of the lipids or triglycerides, that is called lipolysis. Phospholipids, we've talked a lot about phospholipids, and you know it's important for the plasma membrane. Then steroids, they are also type of the lipids. And then eicosanoid. Now carbohydrates. What are the monomers of carbohydrates? What are the monomers?
Monosaccharides.
Yes. Monosaccharides. An example of monosaccharide would be?
Fructose and glucose.
Okay. But you really want to remember glucose because we'll be talking a lot about glucose throughout our physiology. So glucose, fructose, so know the examples. So monosaccharide, example glucose, fructose, even galactose is a monosaccharide. And polysaccharide, of course you can see starch here. Any other polysaccharide we have talked about? We talked about it not too long ago in the lecture. What is that polysaccharide? What is the stored form of glucose in the body? What is it called?
Is it
Glycogen.
glycogen?
Yes, yes. Glycogen is a polysaccharide. It's important from physiology point of view because that's how we maintain the normal blood sugar levels because of glycogen and being available. And here you see that. Okay. Okay. Nucleic acids. What are the what are the monomers of nucleic acids?
Nucleotides.
Nucleotides. Okay, now let's review the DNA and RNA. What is DNA made up of? What is the sugar in DNA?
Deoxyribose.
Deoxyribose. Okay. And then it has a phosphate. And then what are the 4 nitrogen bases in DNA?
Cytosine, guanine, adenine, and thymine.
And how do they pair? What is the pairing?
Adenine pairs with thymine and guanine pairs with cytosine.
Yes. Please know this stuff. This is important. And not just for this class. You will need this information more when you do more of the biochemistry part. And DNA is always double-stranded. RNA. What is the sugar in RNA?
Ribose.
Ribose sugar. What are the nitrogen bases in RNA?
Adenine, guanine, cytosine, and uracil.
Yes. So you are going to replace thymine with uracil. So if you see uracil, that means this strand is a RNA strand. You should be able to construct the RNA strand. If you're given a DNA strand, not or the nitrogen bases on the DNA, and the question asks you that you have to figure out which one is the RNA strand which was built from this set of nitrogen bases in the DNA. You should be able to figure out which one would be the RNA strand. We did that in the class. You will see one question on that. We've talked about adenosine triphosphate. Okay, now the protein. What are the monomers of proteins?
Amino acids.
Yes, amino acids. And when you call it a protein, how many amino acids should be there to call it a protein?
Over 200.
Yes, you have the information here, more than 200 amino acids, only then you will call it a protein. Less than that, you will call it a polypeptide or oligopeptide. I just want you to get familiar with all these terms because you will be using these terms a lot, but you need to know what these terms mean. In general, oligo always means scanty. So wherever you see the prefix oligo, you know we're talking about something which is scanty. And poly means many. Okay, now what are the different structures? What is the primary structure of protein? What is the secondary, tertiary, and quaternary? What is primary?
A linear sequence of amino acids with peptide bonds.
And yes, and that is the raw protein. When the protein just gets manufactured on the ribosomes, that is the primary structure. It's just a linear. All the amino acids connected together by a peptide bond, and it's one linear chain. That is the primary. Then what is secondary? So the primary gets modified. Instead of it may be still linear, but then you add more to it. Either you make it a spiral, or it has the pleats. It's so that would be the secondary. So but it is still one dimensional, or we can say just two dimensional. Then the tertiary is the secondary structure gets further modified and folded, so now it becomes a three-dimensional. But still it is only one kind of protein chain which is further getting folded and refolded. That is tertiary. And then the last one is the quaternary. So what is different here? What is this quaternary structure? You have more than one protein. Till the tertiary structure, it is only one protein which is further getting modified. But when we say quaternary structure, that means now you have different kinds of proteins all connected together. And that is quaternary. You might want to remember hemoglobin is one of the examples, which has 4 protein chains. So that is what is quaternary, when you have more than one protein. We've talked about denaturation. Okay. Any questions in this chapter? Okay, I'll move on to the next one.